Thank you. I will hurry through my presentation so we have time for questions at the end. I'd like to share with you some of the information that we have looked at that maybe can shed some light on some of the comments and questions you've had so far with some data looking at a large body of food nutrition research. My disclosure for past funding, as well as this funding, this project was funded by the International Life Sciences Institute, the scientific assembly of scientific integrity. They had no role in the conduct of the research or the findings. When we talk about research behavior, we've kind of, Cochrane has identified it as having three different components. There's the research integrity, academic integrity that deals with, copying material and things like that, and then responsible conduct. And so this one really looks at one of the 6 fundamental values of research integrity, which is focusing on the responsibility as a researcher. And as a researcher, then, you are responsible for conducting research in the ways that fulfill the professional responsibilities, like as Kathy had mentioned, that are set out by your professional society, by your institution, so that they are relevant and to the public. In other words, what you're trying to do as a researcher is to reduce risk of bias in your research, and that's what we're going to be looking at. We focused our research around the constructs from Cochrane with the 5 risk of bias domains. The selection, in other words, how you put individuals into the study or allocate them to groups if it's a observational study. Performance, how well you actually measured or captured the exposure or conducted the intervention. Detection, how well you measured the outcome that you're of interest. Attrition, whether or not you were able to keep the individuals in the study or throughout the time period, and then how well you reported that research in the actual literature published. The purpose of what we were looking at was to examine the risk of bias domains to explore exactly what kind of shortfalls exist in the body of food and nutrition research. It's nice to talk about the impact of different things, but we really wanted to know where were the shortfalls and the opportunities for improvement. And so in order to do that, we used critical appraisals that had already been completed by 3 different systematic review systems. We looked at the Academy of Nutrition and Dietetics Evidence Analysis Library. We extracted data from the Nutrition Evidence Library from USDA, and we selected AHRQ reports and extracted as much data as was available on those reports that were relative to food and nutrition. Our specific aims were to evaluate the relationships between those risk of bias domains, whether they were met or not met, looking at the different data sources, looking at consistency between ratings given to the risk of bias, between multiple sources, and then finally to evaluate the relationship between those domains and the type of research, publication year, and source of funding. And we looked at that within each data source as well as across all 3 data sources. To give you an idea of the sample, when we polled, there were 4,207 research articles that had been evaluated in the Evidence Analysis Library. There were over 1,100 that were included in the Nutrition Evidence Library, and there were over 900 that were in the AHRQ system. So all told, we had a total of 5,675 research articles that had been evaluated. They're unduplicated. There were 641 that were duplicated in more than one system. It had been evaluated by the EAL, by the AHRQ, or the NEL. To give you an idea of the distribution of the sample, when we looked at the intervention research over here, you'll note that 65% of the trials included under intervention were randomized control trials. However, there were 30% of them that were in non-randomized designs, these designs here. When you looked at the observational, there were 37% prospective cohort and 34% case control. And the other smaller designs are listed there, but those are the bulk of it. When we looked at the type of research design, we noted that intervention, they were almost equally split between intervention and observation. 51% were intervention trials and 48% were observational. The N's are listed there. Between the data sources, we know that AHRQ relied more on intervention studies because that was usually part of their inclusion-exclusion criteria, that they be randomized controlled trial or quasi-controlled trials. The other 2 systems, the EAL and USDA, included more observational trials than an ARC did. To give you an idea of the funding source, this gives you an idea of the government, the combined funder here for intervention trials and the combined funding for observation, government funding for observation and intervention. Now, another way of looking at this is this particular diagram, and this shows you by the combining of funding and what types of studies were included. So here's the intervention studies. With government funding, you'll notice that the predominance of those were randomized controlled trials. Combined funding produced more research than government only, and that included randomized controlled trials and the non-randomized trial and the non-randomized crossover trials. In observation, you'll notice that here the government is the predominant funder, followed by combined funding. Again, prospective cohort, the largest group, followed by cross-sectional and then case control. One thing that's interesting about this is when Lanahan looked at sources of funding in life sciences in general, he had concluded there was about 61% was government funding. When you look, but he didn't include a combined category, he just looked at anything that had government funding. When you look at our data here for nutrition, this body of nutrition and research only had 49% where there was a possibility of government funding. Either government alone, 23%, or 26%, which is combined funding. And combined funding often included government plus some additional funds in funding and sources in addition to that. We looked at a wide variety in publication years from 1930 all the way up to the last one that was included was 2015. And it gives you by data source, it found that the NEL had completed fewer reviews with older research than the other two systems. The categories of years that we used for evaluation are here, pre-1990, 1990 to 2000, 2000 to 2010, and then 2010 on. Now, these are important because you remember that the consort standards that were mentioned earlier, the publication guidelines, started coming out in the late 90s, but they really came into full force in the 2000s. So that was one of the year for the cutoffs, is approximately when those started to go into use. The overall sample in terms of looking at quality ratings, and all of our systems used a 3 quality rating, either positive, neutral, or negative, and this gives you the distribution between, for interventional studies and observational studies. The 41% were positive as opposed to 35 and 24, for observational 45 versus 49 and 46. The key differences in the samples were noted. There is a difference in the research type I mentioned earlier and design, the relevance or the amount of reliance on intervention versus observational, as well as the specific designs within those two categories. There was difference in publication year. NEL had slightly newer data than we had older data sources than some of the others. There was difference in funding source between the three databases. And only EAL and AHRQ had what we call overall quality rating, that plus, minus, and negative. The other reports in NEL only had individual item ratings that we could use for the risk of bias domain. We mapped the items to the risk of bias domains. There were different instruments that were used. The quality criteria checklist was used by the Academy of Nutrition and Dietetics. The research and design implementation and the bias assessment tool were used by the Nutrition Evidence Library. And the AHRQ used a variety of tools, but they customized the instruments to each question sometimes, and sometimes to each report, and sometimes down to the question level. There were individual items that were used on their critical appraisal. The way we conducted the mapping is we had independent researchers go through and rate each item in terms of what domain it would match to, and then we had a consensus discussion of, on what the domain would be if there were any differences. We created a mapping guide that kind of showed us what types of items off of those questions would fall into each of the 5 domains, and those will be published so you can look at those later on. Then we explored the relationships between the risk of bias domain and whether or not they were met. And the first data we just looked at was percent of the items or percent of the risk of bias domain that was listed as being met. Now, what I want you to look at is just the progression here between the selection, performance, and detection. So selection and performance are much lower on intervention research. The last 3 bars on each one are the non-randomized trials. So when you look at those, the randomized trials are going to be on this end, and then the others not meeting or over there. Same thing for observational trials. Here you see that selection, performance, and detection are the ones that are the lowest, providing the greatest opportunity for improvement. The next thing we looked at then was the risk of bias and whether or not those risk of bias domain ratings predicted the overall quality. In other words, if a researcher downgraded it from being a positive to either a neutral or negative, which of those risk of bias domains seemed to have the biggest impact on that decision. So those are listed here, all the significant relationships. The ones that are in bold are the ones that had the largest impact within those that are significant. So for example, downgrading an intervention study to neutral, selection was a key domain. When it went down to downgrading it to negative, you found selection and detection both were very large. For observational studies, again, selection was a key driver for reducing it to neutral. Selection and detection were on for the negative. So across the board, if you look at these, the selection, performance, and detection were significant predictors for reduction for both neutral and negative on both types of research designs. Since we use different instruments and different databases, one of the things that was very important to us was to see if the ratings were similar, if ARC graded a study versus if the Academy graded the study versus if the USDA graded the study. So we looked at them. Within ARC, we mentioned that study could be, or the evaluation occurred at the question level. So within a report, they could have more than one time that they evaluated a research article and they could have scored it differently. They spent a lot of effort customizing their tools to each question that were in the ARC reports. But when we looked at those, there were 841 instances when there were duplicate ratings, but the agreement is very high. This is the percentage that disagreed. So the discrepant ratings went from like 0.6 and the highest was 3% on detection. So even though they spent a lot of effort creating individualized questions, in the end, their evaluation of how good the study was remarkably consistent. Then we looked at, and that's a good thing, yes. Then we looked at the critical appraisals that were included in multiple systems. So between now the EAL system, the ARC system, and the NEL system. And you'll notice here that the sample size, the only one we really have a great deal of confidence in is the overlap between the NEL and the EAL. What we found was surprising to us that was only 10% overlap. Out of those 5,900 articles, there was only 10% of them that had been evaluated by more than one. So on one hand, that's really good news that we're not duplicating efforts and doing the work over and over again in 3 different systems. But it's kind of surprising if you think about the nature of food nutrition research that they wouldn't have used the articles in different questions in the different systems. The good news again was that there was very high agreement between the EAL and NEL when they evaluated the projects, and the CAPA ratings were 0.86 to 0.94. So in summary, there was a small amount of overlap between the different systems, and between system sample size was really only sufficient to be confident in our conclusion that they were similar was between the NEL and the EAL. And for the other comparisons, the sample sizes were quite small. Next, we looked at models that could predict whether or not the risk of bias domain ratings were being met in the critical appraisals. We used a binary logistic regression, and we included funder, the year published in those categories, the research type, whether it's intervention or observation, and then research design. We used 2 models, one model for intervention, one for observation, and separate models for each risk of bias domain. Again, with the funder, the comparator we used was government funding. For category of year, we compared it to the oldest, which is pre-1990, to indicate what was different from 1990 publications. And then the research design for the intervention, we chose our randomized controlled trial as our comparator as the gold standard. And then for the observational, we used the prospective cohort trial. Now this is just kind of a visual to show you when I talk about funding and we say that there's no consistent association between source of funding. The little arrows tell you where there were significant relationships, and those in the box increased the quality, in other words, reduced the risk of bias. So some were, the ones that are not marked reduced it compared to government funding, and the 2 in the box increased it. But you know, you have here, for example, not reported funding. There's 2 instances, both of them in selection. Industry only, there's 1 in selection for intervention, and so forth. So there's not really any consistent thing. You can't say this source of funder across the board is going to produce a low-quality research project. When we looked at research type and design, there's much more to say. We have the non-randomized crossover trials. Here there's the opportunities for that they reduced the, or they had a increased risk of bias, opportunities for improvement here would be a non-randomized controlled trial for the observation in the case studies and in the time series, had the biggest opportunities. If you look publication year, we show very consistent improvement by category of year published. The only exception is performance didn't start improving until after 2000. And the odds ratios for the relationship between publication year are between 1.56 and 6, and the largest one was the reporting category between for the 2010 to 2015, and the odds ratio were 16 and 42. The strengths and limitations of the study, we used existing data that was not originally developed to capture specific risk of bias domains, although they were mapped to them. There was the use of more than one instrument so it could introduce variability. And one of the potential confounders that is unaccounted for in here is the look at the consistency between other parts of the system, like how well the reviewers were trained and so forth. But given the fact that the consistency between the ratings is very high, we don't think this is a serious limitation. The strengths would be the sample size, a large amount of diversity of topics, the diversity in funders, including both intervention and observation research. And then there was one instrument that was used for a large portion of the sample. And it was the first time that the risk of bias domains were used to identify opportunities for improvement in a body of research. Our overall conclusions is there's little overlap between the three systems, very high consistency in the ratings. Publication year and the research design were more consistent predictors of risk of bias. When it was different, government funding usually predicts less likely to meet the risk of bias criteria, in other words, higher risk of bias, but there was no consistent pattern for the funders. And across the board, the greatest opportunity for improvement for food nutrition research appears to be in selection, performance, and detection domains. And just to kind of remind you what that means, if you're talking about selection, this is the recruitment and allocation to groups. Performance is really looking at the management and measurement of the intervention exposures or the independent variables. And then detection is how well you measure the outcomes or the independent variables. And listed below here are the types of research designs that contributed to those opportunities for improvement. The non-randomized trials, and then all designs except for case control were lower than prospective cohort. Non-randomized high performance and non-randomized for controlled trials. Now this one you can understand the non-randomized and having a little opportunity to improve that, but there should be room for improvement in performance and detection in the non-randomized trials. So potential implications for researchers, and I think it goes along with Kathy's comments and also Russell's comments. Our suggestion is to develop nutrition-specific extension to the reporting guidelines for the various types of research study designs. We have Consort for trials, you've got Strobe for observational, you've got a ton of reporting guidelines that tell you, have you done your research well? But we don't have any specific extensions for nutrition, and there are some unique considerations that need to be put on the table to be addressed. When I went to Equator and looked at the guidelines, herbal medicine has their own extension, pharmacy has their own extension, and we really, really need to have a nutrition extension. So I'm going to encourage the organizations that are moving forward to look not necessarily at developing new guidelines, but extensions for the existing guidelines that address nutrition. And the classic example would be how well they characterize background diet, how well they characterize what are in supplements. I mean, or if you're looking at energy, how well they measured the energy that they're talking about. So I mean, those kinds of things are very specific to our field. So it's not reinventing the whole wheel. It's just adding some specific questions to it that will make our interpretation of how well the research is done better. And then we did not find a significant impact of funding. So that question, we looked at the process of research, how well the research was done. So if somebody is out there looking for a new research project, then the next project would be to go back and look at the outcomes in terms of how the risk of bias and domains, if there's any relationship between the direction of the outcomes. But we looked at the process. And then, consistent with what Kathy was saying, looking at enhancing education training for researchers, specifically on those three domains, selection, performance, and detection, and specifically on those research designs. If you're going to use those designs, what can you do to maximize it? Because as you mentioned, it's a trade-off between can you afford to do the other types of research designs, oftentimes. And then organizational changes for societies or academic organizations, the policy and procedures that you were talking about for scientific integrity for both of those things, enhancing the information specific to those 3 domains, re-examining the research designs that seem to identify the most ways to contribute to those domains, and then continued commitment to funding disclosure. Remember on that slide with funding, there was a large percentage of them that had no funding reported. Have you ever done research without any funding? I mean, somebody paid your salary most times, somebody paid your time, so it's unclear how there could be that many studies that were done with no reporting. But the interesting thing is, as we have become more committed to conflict of interest, over time, that percentage went down. So we are getting better at, being full disclosure of all the funding sources when we do that. So the last one is continued commitment to a full funding disclosure. If somebody pays your salary, that's funding. And then if I was just dreaming, I would say intervention to increase the knowledge or skills and attitudes, specifically with the modules on those 3 domains. And then I'd love to have an ongoing metric, and my suggestion would be to look at the journals that are using the existing guidelines, the checklists, because they're completed during the peer review process for many of the journals. Figure out a way where you capture that information. You can capture that information when it's turned back by the reviewers by making that automatic, and then you can track things over time. You can look at those things that were published, those things that weren't published. It's useful to authors to tell them, this is the most common reason that your journal article is not accepted, or no, you wouldn't say that. You would say that there's a high prevalence of this domain being associated with not being published. And then you can tell what things are published and is our body of research improving over time. So that'd be my dream. Acknowledgements, co-authors Scott Parrott from Rutgers, Pat Splett, Mei Chung from Tufts, Deepa Handu from the Academy of Nutrition and Dietetics. Special thank you to Joanne Spahn from Nutrition Evidence Library, the researchers at grad students, and of course, ILSI North America's Scientific Integrity for the support. And this has been accepted for PLOS One. We really thought we were going to get the email last week giving us a publication date, but I haven't gotten it yet. So,
thank you very much. We have time for a couple of questions for Dr. Myers.
Andrew Brown, Indiana University. I had a hard time keeping up with all that because that's a lot of data, but I'm happy to hear that we'll be able to read it soon. One point I wanted to echo of yours was about the guideline creation, because somebody did try to make a separate guideline for grade, particularly for nutrition research, and kind of reinvented the wheel, and it doesn't seem to uphold a lot of the standards that the community holds for grading. So if we don't take it, somebody else will do it, and it'll become a de facto standard. For the funding, one thing that I thought was interesting, this is just a brief comment about that, but I see and hear a lot where people will say the funder had no role in the research, but in fact, the fact that it was funded in some way is an indication that they approved it. So I think, there's a broader conversation of how we do those disclosures. But the question I had for you in particular was about the risk of bias domains, and whenever I try to assess risk of bias domains, most of it is just missing. So we can't say whether it's high or low risk, it's just missing risk of bias for the original study. They didn't report whether or not they randomized. They didn't report allocation concealment. And did you see a lot of that in your work as well?
When you go back to the slide that had the percent met, so when these here. So the way it was coded is if they could document, or if the article documented that it met it, it was coded as a yes. So this is percent of risk met. If it was missing, or it was unclear or not, it was a 0.
Okay.
So this is telling you what percent we knew met the criteria. So the other 3 were collapsed into the no.
Great. Thank you very much.
Mm-hmm.
Okay, last question for you.
Sorry.
That's okay.
This is Leela Saldana. I actually have a comment in response to something you said at the very end, and I do have a question. My general comment is, if you're going to come up with some kind of a checklist, and this is a challenge you face with some of the consort statements, is they're way too detailed, and you really can't use them to evaluate a paper. So I think you're going to come up with something that needs to be reporting and for grading and evaluation, and that should be very brief on what's the minimum requirement that a study should report. But my question was, when I listened to all three presentations, when we talk about funding sources, we talk about dollars in funding, but I didn't hear from anyone where the test materials were donated by someone. And I think sometimes that has a bigger bias in the sense of who provided the study material. Is it a commercial product that's on the market or something's being tested? And I've seen some challenges with that, and I didn't know that was being considered in all of this.
That would be reported as a source of funding. If there was anything that was donated, that's counted as a source of funding.
So that was
Taken
into consideration.
That would be
in
in
your back- okay,
consideration.
gotcha,
So
okay.
for example, if it was a government-funded and there was product donated, that would be combined funding that had industry and government.
That was not clear from the presentation.
No,
then?
it wasn't. It wouldn't have
Okay,
been.
okay.
Thank you for asking.
Okay, I think we'll wrap up the session here. Would you please join with me in thanking all of the speakers for excellent presentations? And on behalf of ASN and the Canadian Nutrition Society, I want to thank you all for attending this meeting. Thank you very much.